going to cover some of the research discoveries from 2016 from the Dr. Paul H. DrAlan Research Fellowship Endowment. We will cover in capsule many of the detections of spirochetes in patients who submit their specimens for voluntary participation in our research program. So these images are some of the images that we see in people who have no detectable antibody. They're seronegative. Blood and spinal fluid testing with DNA probes for Borrelia burgdorferi show that the spirochetes can be detected in some of the patients who submit their blood and spinal fluid to us. And these are examples of positive spirochetes in people who have a negative blood testing result with ELISA and Western blot. Miyamotoi Borrelia spirochetes can also be detected in blood and spinal fluid from seronegative patients. Of course, there is no serology for Miyamotoi antibody detection in patients who have this infection. It's a relapsing fever Borrelia, and it has a worldwide distribution. So detection of spirochetes here is no surprise. The blood tests are not contributory in Miyamotoi disease. Borrelia species of the Borrelia group SL burgdorferi and Miyamotoi group SL are also detected in autopsy tissues, and we can examine autopsy slides with the DNA probes and show that Borrelia spirochetes are present in these tissues in select cases. Brain tumor studies under the DrAlan Foundation initiative, glioblastoma multiforme, very highly malignant tumor in the brain. In 5 cases that we tested contained the DNA of Borrelia burgdorferi. The reason that we tested glioblastoma multiforme is the notion which was originally formulated by Bonnie Bennett in the United States, that glioblastomas seem to appear in increased percentage of patients with chronic Lyme disease, and she wondered whether there might be a connection. The DNA probes that react here with the tumor cells show that there is a very good connection, and the 5 cases we've studied, the tumor cells do light up. So the DNA of Borrelia burgdorferi may corrupt the benign glial cell DNA and promote the formation of this highly malignant tumor. Glioblastoma killed George Gershwin. It killed Senator Edward M. Kennedy. It killed many, many people in the public eye, and it has a 100% fatality rate. Lewy body dementia is one of our other initiatives, and we've studied diffuse cortical Lewy body disease. We have found that the worms with their endosymbiont Borrelia, that means the Borrelia that lives inside of the nematode parasites, are detectable in the brain, and we think this is one of the neural larva migrans illnesses, migrating worms through the brain, causing tissue injury and dementia. The Borrelia spirochetes partner with the worm in producing tissue injury. You can see the worms here in white, and the red background is the stain for the Lewy body, which is here a red stain with an antibody to the alpha-synuclein entity which forms the Lewy body. It's a toxic protein like amyloid in the brain of Lewy body patients. Chronic Lyme borreliosis is chronic because of biofilms, and biofilms can be found in tissue, biofilms of Borrelia in circulating blood. To the right there is a cluster of Miyamotoi Borrelia in the blood, and this is the colony, community of the Borrelia surrounded by their own matrix, which is DNA and RNA and protein. And to the left is a amyloid-coated biofilm in the blood. These are very large. The red cells are about 7 microns in size. The biofilms may be up to 100 microns. And if the amyloid, which confers rigidity on anything that it coats, hits a capillary bed, it will cause tissue death or tissue injury. This may be one of the reasons why patients with chronic Lyme complain of total body pain. It's not psychiatric. It is due to embolic amyloid-coated biofilms. Alzheimer's disease amyloid plaques or biofilm communities. I covered this last year, so I'm not going to repeat that. The biofilm communities, again, are coated with amyloid. Amyloid is an antibacterial agent. So Alzheimer's disease-coated amyloid plaques, biofilms living inside the amyloid plaques in Alzheimer's disease. These biofilms are Borrelia biofilms. I'm going to spend most of the time here today talking about multiple sclerosis, and we have discovered, we're astounded to discover this, that multiple sclerosis is a parasitic infestation in the nervous system. We call it neural larva migrans. The worms migrate through the brain, and the worms lay eggs. The eggs hatch and larval forms grow up to adult forms. Mating adult pairs produce more eggs. All of these are detectable in the spinal fluid. If you look for them in the brain, you may be very frustrated because it may take several hundred to almost 1,000 sections of cutting brain tissue to find one worm. That's because the worm migrates and creates a storm of injury in its wake, like the tail of a comet. And so if you look at the injured tissue, the worm has already moved on, most likely, and you may not find the worm. It's also possible that the worm may have died, and that the worm no longer has the shape that you expect to find in a living worm. These two pictures, low power to the left, the blue, shows a coiled worm of the nematode type in spinal fluid of multiple sclerosis, and to the right, the end of the worm showing the mouth parts and some little hooks. This a worm, of course, in the brain, an adult worm, and does not belong there. So the focus of this lecture, multiple sclerosis, is a neural larva migrans. That means in the nervous system, migrating larval forms and adult forms of parasitic worms migrate through the brain and cause the multiple sclerosis illness. This discovery in January 2016 happened when we received a case from Oregon, an 80-year-old man, his past medical history positive for chronic Lyme disease of 20 years duration or more, and positive for multiple sclerosis for the last 10 years of his life. After his death, the family asked us to do special studies to try to find evidence of Borrelia in the brain and spinal fluid. The autopsy provided us with cerebrospinal fluid and whole brain. I won't get into the whole brain, but the cerebrospinal fluid is shown here in the image, and that is not healthy cerebrospinal fluid. Cerebrospinal fluid should be free of anything that you could see with the naked eye. This is spinal fluid that has been fixed in alcohol, and you can see that there's a chalky white haze. This is not normal. It should be transparent. There should be nothing in there other than ions and maybe a tiny bit of protein. You can also see that there are rounded things in the spinal fluid that are almost like little granules. And then at 6 o'clock you can see something that looks like a worm. Well, it is a worm. It's an adult worm. And these granular concretions are portions of the worm body or worm eggs that you can see with a naked eye, not even using a microscope. Of course, we use a microscope to be sure that what we see with a naked eye is also seen under the microscope. So the Rocky Mountain Multiple Sclerosis Brain Bank at the University of Colorado School of Medicine, Boulder, Colorado, has been key in our receipt of specimens of spinal fluid from multiple sclerosis patients. Each of their cases is carefully studied with pathology, and there we know that each one is in fact multiple sclerosis by pathology exam of the brain. The spinal fluid was what we started with and what we finished with, and the spinal fluid then with DNA probes for fish study to attempt to detect Borrelia burgdorferi, we did find Borrelia in the spinal fluid, but we found many more worms than did we find Borrelia. inside of each of the worms, with careful examination with the DNA probes for Borrelia, we can find Borrelia spirochetes hiding inside the body of the worm. So they're called endosymbionts. They live inside the worm, and they don't cause any harm to the worm. But when the worm dies, the Borrelia are released, and then they cause trouble in the host. So it's like a Trojan horse. The worm itself is an assault on the body, and the Borrelia inside of the worm, when they're released, also assault the body and cause tissue injury. The two have an unholy alliance and cause a condition, multiple sclerosis, and other conditions in the brain. This unexpected discovery that spinal fluid contains many nematode parasites was completely serendipitous. And once again, we can see the chalky appearance. When you fix the spinal fluid from a multiple sclerosis patient, if it looks like this, until proven otherwise, those are worms in the spinal fluid. Unfortunately today, microscopic examination and making smears as we did is not routine in any laboratory, in any hospital, or any medical school in the world. So we did the first work, and we were rewarded for our curiosity. Additional spinal fluids in the multiple sclerosis brain bank after we got our first case produced 9 more cerebrospinal fluids for multiple sclerosis patients. All of them were positive, 9 of 9 or 10 of 10 total positive for nematode parasites. The nematode species are probably plural, so there's probably more than one species of nematode in the spinal fluid. We're working on trying to establish most likely species based on the egg morphology, the morphology of the larvae, and the morphology of the worms. We know that they're not segmented, so they're nematodes, but there are many, many nematodes. Additional studies, DNA probes for Borrelia spirochetes, positive. Worms contain in their interior milieu spirochetes, Borrelia spirochetes. And in this model, this mimics the symbiont relationship of microbes, pathogens in other nematodes, the nematodes that cause river blindness. So endosymbionts are microbes that do not harm the worm, but they live inside of the worm. There are also examples of endosymbiont microbes inside of amoebae, and of course inside of ticks we know there are many microbes, and those are actually in the definition endosymbionts. The bugs don't harm the tick, but they harm the person who gets the bugs into their system from the tick bite. So worms, amoeba, and ticks all can carry endosymbionts. Endosymbiont model is a poster child. The river blindness model, the worm here, a roundworm called Onchocerca volvulus, carries endosymbiont Wolbachia species. And in this special case, I want to give great praise and a notice of very, very keen insight to Elena Cook, who helps us at the foundation and who instructed me on the model of Onchocerca volvulus with endosymbiont Wolbachia. Wolbachia, a very well-established endosymbiont inside the river blindness worm. And the two have an unholy alliance, and they cause blindness. The President Jimmy Carter Foundation and the Gates Foundation have partnered to eradicate river blindness from the face of the earth, and they're very close to their goal. So endosymbionts, the good is that the endosymbiont microbe stays inside of the worm. It never escapes to the outside until the worm dies. So that's the good. And they help each other. The microbe in the worm helps the worm, and the worm helps the microbe, and the two exchange DNA. And there's DNA transfer between the Borrelia and the worm, just as there is DNA transfer between Wolbachia and nematode worms in the river blindness model. So they help each other. It's not a savory kind of a good relationship, because ultimately the worm dies, and so we get to the bad and the ugly, the good, bad, and ugly. The endosymbionts are released from the worm when the worm dies, and the microbe is unleashed and produces disease in mammals. So endosymbionts, the good, the bad, the ugly, they have the ability to modify the DNA of the Borrelia to the worm, and the worm to the Borrelia, just like Wolbachia can modify the DNA of the worm that holds it, and the parasite worm can modify the DNA of Borrelia. And they modify in such a way that they enhance the pathogenicity of the worm and probably enhance the pathogenicity of the microbe. In our case, it's Borrelia and multiple sclerosis. River blindness, it's Wolbachia microbes, enhanced pathogenicity. So history of worm parasites in spinal fluid goes back to 1880. And the veterinary literature is very instructive. They have told us about farm animals that contracted worm parasites, and they wound up in the spinal fluid, and then the brain was injured. The animals may have died, or they may have been permanently neurologically damaged. So brain injuries in this model are very noteworthy because it's myelin and neural tissue that is destroyed. That's exactly what happens in multiple sclerosis. The migrating worm in the brain is also very instructive because multiple sclerosis has a migratory pattern in the brain. It flares up in one area and then calms down, and then flares up in another area and calms down. And there is a migratory pattern of multiple sclerosis injuries, and it's very, very instructive that the worm's migration through the brain recapitulates the multiple sclerosis injuries in the brain. So that happens in the animals and the nematode transmitted diseases which cause brain injuries and myelin loss and paralysis. And in the years from 1880 to 1950, many papers were written by the veterinarians about worms in India and in the Orient that caused permanent injury or death in farm animals that looked exactly like multiple sclerosis, loss of myelin. Now, the renowned parasitologist, Dr. Innes, a member of the Royal Society, London, and Dr. Shoho, a veterinarian, a very esteemed researcher from Tokyo, Japan, in the year 1952 wrote a paper, Nematodes, Nervous System Disease, and Neurotrophic Virus Infection. And their paper should be read in its entirety. It's about 3 pages, but it's just rich with beautiful language and great discussion of the way the nematode causes injury to the nervous system. One noteworthy thing is that in 1952, they were studying brain, and they were finding the worm in brain, and they were finding myelin loss, just like multiple sclerosis, but it never occurred to them to study the spinal fluid in the animal. Another paper written by Dr. Ennis and Dr. Shoho is cerebrospinal fluid nematodiasis. That's a big medical word for worms in the spinal fluid. And this caused encephalomyelomalacia. Malacia means softening. So if you have osteomalacia, your bones are soft. If you have malacia of the brain, your brain is soft. It should be rubbery, and it should bounce back when you push it with your finger. When myelin is lost, you push on the area of multiple sclerosis damage, loss of myelin, and the finger doesn't bounce back. So malacia means softening. And the worm that they were interested in studying is Soteria digitata. They also said in their title that a disease which may occur in man. So they were really prescient in their thinking about animals as a model for human disease. And they were thinking about multiple sclerosis in 1950. I was born in 1948. I'm 68 years old this year, and nobody's touched this paper in my entire lifetime except me. So, the animal model lumbar paralysis from the year 1880 by Griffith Evans was called Sura in Ceylon and India. Pathology was malacia, softening of the brain. Myelin's destroyed, neurons are destroyed, and the wandering worm causes a storm of trouble as it moves through the brain. Weakness, incoordination, paralysis, all these things happen in multiple sclerosis. They also happen in animals. There may be death that happens in multiple sclerosis, or there may be permanent neurologic injuries, as happens in multiple sclerosis and in the animals with this condition. Sura, S-U-R-R-A. Animal autopsies, liquefaction of the brain and spinal cord in patchy areas where the worm has passed through. And that's what happens in multiple sclerosis. There's liquefaction and loss of myelin and loss of neural tissue. So in the autopsy, looking for the worm, it took hundreds and hundreds of slides to find one worm, because the worm causes damage and then moves on. So if you study the area of softening and damage and liquefaction, you're going to miss the worm most often. So in 1952, they really worked very hard to prove that there was a worm there, and hundreds of slides were taken from the animal brains. In 2016, our discovery was it's much easier to find worms in the spinal fluid in such cases, and it's like shooting fish in a barrel. Spinal fluid was not examined in the animal autopsies or in human autopsies of multiple sclerosis until 2016, when the DrAlan Foundation began its MS spinal fluid incentive, and we are continuing with our work. We have not found a case of MS that does not have worms. Multiple sclerosis-like illnesses in the veterinary model, farm animals in India, elsewhere in the Orient. The work of Innes and his colleagues really set the stage for their prediction of a hypothesis that the animal model could be a precedent for human MS studies. And they were absolutely right in 1950. Nobody picked up the ball and ran with it after 1950. So once again, the multiple sclerosis spinal fluid looking very unhealthy, chalky. You can see a worm, you can see rounded things. They might be eggs, it might be worm debris, it might be worm poop. They do not belong in spinal fluid, and they all exert a toxic effect on the brain. If you bleed into the space where the spinal fluid is, the blood in and of itself will damage the brain by causing intense spasm of the blood vessels, and you will have a stroke-like event. So blood in the spinal fluid regions of the brain can cause injury because it produces vascular spasm. The brain is very, very picky about what goes on in the spinal fluid. And so you can only imagine what worms in the spinal fluid will do. It's a very, very unsavory scenario. These are the worms of a case of multiple sclerosis from the Boulder, Colorado MS Brain Bank. You can see it's almost like a starfish. And you can see the size grid. Those are each division, spaces between the two, small division, 10 microns. 10 microns is a little bigger than a red blood cell. The worms again, and in a cluster. More worms, an egg, actually a fertilized egg, a mating pair of worms. You can see the male is on the top and the female is on the bottom. Those are in red, and then the blue is the background. This is a segment broken off from an adult worm, and you can see the intestinal tract that's running a straight line from the top of the worm to the bottom segment. And this is a zygote, developing embryo. So 10 patients with multiple sclerosis, 10 spinal fluids contain nematode, adults, larvae, and eggs. Conclusion: nematode parasites and spinal fluid in multiple sclerosis. Expected, you'll see a chalky appearance after fixation. That in and of itself tells you there are worms there, because nothing else causes the spinal fluid to look chalky except junk. And you see the debris, and sometimes you'll see the worms with the naked eye. They're easily identifiable. Dark field microscopy is recommended to see them. You don't need to stain them. Just put them under the microscope and start looking. The dark field should be an oil dark field. There are dry dark fields and oil dark fields. Oil dark fields give you higher resolution and allow you to see smaller and smaller things. So oil dark field microscopy is what I use. Implications for treatment: nematode worms, parasites, can be eradicated with anti-helminthic medications. That's anti-worm medications. Every veterinarian prescribes these every day for doggy heartworm. The medications are safe, effective, and cheap. Now there is a cautionary note, and that's in the next slide. Cautionary note: careful, very careful, dose escalation, which means you start out with a low dose and you gradually, very slowly bring the dose up to avoid Herxheimer-like events, which may be fatal in the brain. You don't want to have a Herxheimer in the brain. Remember, the brain blood vessels can go into spasm when there is not peace in the valley. Corticosteroids They are medications to reduce inflammation. Should be given with worm meds to protect against worm kill flares. So be very careful with your anti-worm medicine if you have multiple sclerosis and worms in your spinal fluid. And your physician should be very careful. That means you should not go out to the store and get anti-worm medicine from your vet and take it without physician supervision. We don't want any people who have multiple sclerosis to have adverse or fatal outcomes. So this is the end, and I thank you very much for your kind attention. I'm going to take questions from the audience, try to answer them, and I hope that you have found this as interesting as we do in this year 2016. My goodness, 60-something years after the paper by Innes and Joho. Thank you very much.